Hi everyone, and welcome to our module on fatty acids. Before we talk about fatty acids, let's define the broad term of lipids. Lipids are structures that are made up mostly of carbon and hydrogen, and in general they are usually not very soluble in water, and there are many types of molecules which fall under the heading of lipids. This includes fatty acids, which we'll talk about in this module. This also includes molecules called triacylglycerol molecules, or triglycerides, which we'll see a picture of on the next slide. And then other structures that fall under the heading of lipids include cholesterol molecules, phospholipids, steroids, and glycolipids, and you may be familiar with all of these from studying cell biology and biochemistry. On the top left side of the screen here, I've shown the structure of a fatty acid. You can see that one end of the molecule has a COOH group, and this end can give off a hydrogen, and therefore that makes this an acid. However, most of the molecule is composed of a long chain of carbons and hydrogens. There may be single bonds or double bonds, as I've shown here, but importantly, there are no oxygen molecules, no nitrogen molecules. There are just carbon and hydrogen molecules, and that means this portion, which is the dominant portion, is not going to be soluble in water. On the top right side of the screen is a molecule of glycerol. It's a short carbon molecule with several OH groups attached, and glycerol can bind to 3 fatty acids to make a molecule of triacylglycerol, which most people call a triglyceride. You can see that it's got the glycerol molecule here attached to 3 different fatty acids here. It's called triacylglycerol because it's got acyl groups which contain these double bonded oxygens, and it has 3 of them, therefore it's triacylglycerol. But as I said before, most people call this a triglyceride. And a triglyceride is a way of carrying 3 fatty acids around connected to a molecule of glycerol. The reason I showed you those chemical structures is because the 2 main ways by which fatty acids are transported through the body is either as free fatty acids or as triglyceride molecules. Most of the lipids that you eat in a meal are degraded to free fatty acids in the intestines, and then the enterocytes convert the fatty acids into molecules of triacylglycerol or triglycerides. These are then packaged into chylomicrons, which carry them through the plasma, and then when the triglycerides reach the tissues where they're going to be used or stored, they are degraded back into free fatty acids. There's a very important enzyme called lipoprotein lipase, which is found on the endothelial surfaces of capillaries, and it can degrade the triglyceride molecules back into 3 molecules of free fatty acids. And this enzyme is abundant in adipocytes and muscle tissues, and these are the places where fatty acids are often used. Before we move on and talk about fatty acid synthesis and breakdown, let's define some vocabulary. When we say that a fat or a fatty acid is saturated, we mean it contains no double bonds. This is a picture of a saturated fatty acid here because all the bonds are single. And we say it's saturated because it's saturated with hydrogen, and therefore all the bonds are single. Saturated fats are usually solid at room temperature, and these are the bad fats that often raise people's LDL or bad cholesterol. We'll talk more about that in another module. Unsaturated fats, on the other hand, contain at least 1 double bond. So if we had 1 double bond here, this would no longer be a saturated fat, and it would be an unsaturated fat. We call it monounsaturated if there is just 1 double bond, and we call it polyunsaturated if there is more than 1 double bond. Nutritionists often advise patients to avoid trans fats. So what do they mean by that? Well, double bonds in unsaturated fatty acids can be either in the trans or the cis configuration. This has to do with the orientation of the double bond in 3-dimensional space. Most naturally occurring fats have the cis configuration, but you can get lots of trans double bonds when you use a food processing method called partial hydrogenation. And these trans fats have been shown to increase LDL, which is the bad cholesterol, and lower HDL, which is the good cholesterol. Another type of fatty acid which is good for you is called an omega-3 fatty acid. This is a type of polyunsaturated fatty acid. If you look at the fatty acid on the bottom of the screen, this is an example of an omega-3 fatty acid. The carboxylic acid end of the fatty acid is called the alpha end, and the other end is called the omega end. So if there is a double bond, 3 carbons from the omega end, as there is here, this will be called an omega-3 fatty acid. Lots of these are found in fish oil, and consumption of high amounts of these have been shown to lower triglyceride levels. Now let's move ahead and talk about fatty acid metabolism. There are basically 3 topics under the heading of fatty acid metabolism that we'll discuss in this module. First, we'll talk about fatty acid synthesis. So fatty acids are synthesized largely in the liver and also in the mammary glands. There's a small amount that occurs in the adipose tissue, but mostly adipose tissue is where fatty acids are stored, not where they are synthesized. And excess carbohydrates in the body and proteins can all be funneled towards fatty acid production, as we will see. Next, we'll talk about fatty acid storage. It's largely stored in adipose tissue, as you likely know, and it's mostly stored as triglycerides. And then finally, we'll talk about the process of breaking down fatty acids. Fatty acids contain a lot of potential energy, and the process of beta oxidation can convert fatty acids into acetyl-CoA, which can then enter the TCA cycle to generate ATP for cells. We'll start by talking about fatty acid synthesis. So in high energy states like the fed state, after you've eaten a large meal, there is a lot of acetyl-CoA inside of cells. Carbohydrates are converted to acetyl-CoA, and even some proteins are metabolized into acetyl-CoA. And all that acetyl-CoA can enter the TCA cycle and generate lots of ATP. As a result of the high level of ATP, there will be inhibition of the enzyme isocitrate dehydrogenase in the TCA cycle, and this will result in a high citrate level inside of cells. If you look at the diagram on the bottom of the screen, I've shown you the first step of the TCA cycle. Oxaloacetate is combined with acetyl-CoA by the enzyme citrate synthase to generate citrate, and then citrate is further metabolized by the enzyme isocitrate dehydrogenase. So when the ATP level gets high, it will inhibit this enzyme. Therefore, the level of citrate will begin to rise. And citrate is used as an energy marker inside of cells. When its level gets high, it means that there's lots of excess energy around, and therefore, as we will see, citrate will then be diverted into the synthesis of fatty acids. Step 1 in the synthesis of fatty acids is to move citrate to the cytosol via a transporter called the citrate shuttle. As we will see, acetyl-CoA molecules can be used to synthesize fatty acids, but there's a problem, and that is that acetyl-CoA cannot exit the mitochondria to enter the cytosol where fatty acid synthesis takes place. For this reason, those acetyl-CoA carbons will be combined with oxaloacetate into molecules of citrate, and the level of citrate inside the mitochondria will rise in high energy states as we just described. Citrate can then be shuttled out into the cytosol to participate in fatty acid synthesis, and the key point here is that acetyl-CoA cannot cross the membrane. That's why citrate has to be used. This should make sense to you if you've watched the module on glycolysis, because as we discussed in that module, citrate is an inhibitor of glycolysis via the enzyme phosphofructokinase 1. Citrate is used as a marker of what's going on inside the mitochondria. Acetyl-CoA cannot get out of the mitochondria, but citrate can. So when citrate levels build up, they will enter the cytosol, inhibit glycolysis, and then be diverted into fatty acid synthesis. The next step is to convert citrate into acetyl-CoA. The enzyme that carries out this step is called ATP citrate lyase. As the name implies, it depends upon ATP. And the net result of these first two steps is that excess acetyl-CoA will be moved to the cytosol using citrate as a shuttle. Step 3, once we have the acetyl-CoA in the cytosol where we want it, is to convert the acetyl-CoA to malonyl-CoA. This is the rate-limiting step in fatty acid synthesis, and just like the rate-limiting steps in other pathways, this is where most of the regulation occurs. On the bottom left side of the screen is the structure of acetyl-CoA, and on the right side of the screen is malonyl-CoA. And you can see that malonyl-CoA is larger by one carbon molecule, and that carbon comes from carbon dioxide, and therefore the enzyme that catalyzes this step is appropriately called acetyl-CoA carboxylase. And just like many other carboxylase enzymes, acetyl-CoA carboxylase uses biotin as a cofactor. We'll talk more about that on the next slide. There is some very important regulation that occurs at this step. I told you that when citrate levels are high, it means the energy content of a cell is high, and therefore citrate will activate this step to stimulate fatty acid synthesis. Insulin levels are also high after a meal, and that will stimulate this step so that fatty acids will be synthesized. And then glucagon and epinephrine have the reverse effect. They will inhibit this enzyme. And then finally, malonyl-CoA is a very important tie-in with beta oxidation, which is the breakdown of fatty acids. Malonyl-CoA inhibits beta oxidation so that you don't have fatty acid synthesis and breakdown going on at the same time. I've shown a review article by Daniel Foster on the bottom of the screen here. He was one of the first people to show that malonyl-CoA inhibits beta oxidation. And this review article is a nice way to make some of these concepts of fatty acid metabolism stick in your mind by reading about how they were discovered. Let's briefly review the vitamin biotin, which is also called vitamin B7. It's an important cofactor for carboxylation enzymes. These enzymes all add one-carbon groups to molecules via carbon dioxide. In the gluconeogenesis module, I talk about pyruvate carboxylase. We're talking about acetyl-CoA carboxylase now. And later in this module, we'll talk about propionyl carboxylase, which is important for odd-chain fatty acids. Step 4, once we have created molecules of malonyl-CoA, is synthesis of a structure called palmitate. The enzyme that catalyzes this step is called fatty acid synthase. It basically combines carbons from acetyl-CoA and malonyl-CoA to make a large 16-carbon fatty acid called palmitate. This is the structure on the bottom of the screen here. Fatty acid synthase is a multifunctional enzyme that catalyzes a complicated series of reactions to create palmitate. You don't need to know all of that, but what you should be aware of is that fatty acid synthase uses NADPH, which comes from the HMP shunt, and therefore one of the many roles of NADPH is to assist in fatty acid synthesis. Once palmitate has been synthesized, it can be modified to create other fatty acids that the body may need, and then those fatty acids can be used by tissues based on their individual needs. When there are excess fatty acids present in the body, they will be converted into triacylglycerols, and triacylglycerols are the molecules that are stored inside of adipose tissue. So now we have talked about how cells can start with citrate and synthesize fatty acids from palmitate. We've also talked about how those fatty acids can be converted into triglycerides and stored in adipose tissue. But now let's talk about how our body breaks down those triglycerides in adipose tissues when it wants to use those carbon structures for energy. And the most important enzyme here is an enzyme called hormone-sensitive lipase. It removes fatty acids from triglyceride molecules in fat cells, and it's activated by glucagon and epinephrine so that when these hormone levels increase, they will stimulate the breakdown of triglycerides into fatty acids that can be used by cells in the body. So this is the structure of triacylglycerol, or a triglyceride, on the screen here, and this is the structure in which fatty acids are stored inside of our fat cells. When hormone-sensitive lipase is activated, it will release free fatty acids into the plasma, and it will also release molecules of glycerol. Basically, it will lyse the structure right here and let the molecule of glycerol go free and send the free fatty acids out into the bloodstream. We'll talk about what happens to those free fatty acids in a minute, but first let's talk about the fate of those glycerol molecules that come from triglycerides. Glycerol molecules can travel to the liver, where there's an enzyme called glycerol kinase that can convert glycerol into glycerol 3-phosphate. Glycerol 3-phosphate can then be converted into dihydroxyacetone phosphate, which you may recall is an intermediate in the glycolysis pathway. This means that those glycerol molecules can go to the liver and then be used for energy or be converted back into glucose via the pathway of gluconeogenesis. So glycerol molecules are soluble in water and they can be easily transported through the plasma to the liver. But what about fatty acids? Remember, I told you fatty acids are not soluble in water. Therefore, they need help to be transferred through the plasma, and they get that help from albumin. They're bound to albumin and they're transferred from adipocytes to tissues that are going to use them as a source of energy. There are two important tissues that do not use fatty acids for energy that you should always remember, especially for step 1. Red blood cells conduct only glycolysis. They lack mitochondria, and they cannot conduct other metabolic pathways, including beta oxidation of fatty acids. And then finally, the brain does not use fatty acids as a source of energy. It uses glucose and ketone bodies only. The reasons for this are poorly understood. It used to be believed that fatty acids could not cross the blood-brain barrier. It now appears that it is more complex than that, but in any event, evolutionarily, our brain does not have the ability to metabolize fatty acids. Once fatty acids travel via albumin to the tissues where they are going to be used as a source of energy, they are further metabolized by a process called beta oxidation. This process is the stepwise removal of 2 carbon units from the long-chain fatty acid molecule, and it produces acetyl-CoA, which can enter the TCA cycle, also NADH and FADH2, which can enter electron transport. And as you can imagine, with the fatty acid being such a long molecule, it can produce lots and lots of acetyl-CoAs, much more than can be produced from a single molecule of glucose. The first step in beta oxidation involves conversion of fatty acid molecules into molecules of fatty acyl-CoA. On the left side of the screen is the generic structure of a fatty acid. It has a long chain of carbons and hydrogens represented by the letter R here, attached to a carboxylic acid group. There is an enzyme called long-chain fatty acyl-CoA synthetase, which will convert this molecule into a molecule of fatty acyl-CoA. Basically, it leaves the long chain of carbons and hydrogens intact. It also leaves the carbon double bonded to an oxygen intact, but then it adds a molecule of coenzyme A. Obviously, this process requires coenzyme A, and it also requires ATP. Step 2 is to transport the fatty acyl-CoA molecule from the cytosol where it was formed to the inner mitochondria where the remaining steps of beta oxidation will occur. And this transport involves something called the carnitine shuttle. This is the structure of the molecule carnitine on the screen here. It's a molecule that's found in nature. You can consume it in your diet. It can also be synthesized by the body from the amino acids lysine and methionine. However, only the liver and kidney can synthesize carnitine de novo. The muscle and heart depend on dietary sources or carnitine synthesized by other tissues. So now let's walk through the process whereby the carnitine shuttle moves molecules of fatty acyl-CoA from the cytosol to the mitochondrial matrix where they can undergo the next steps of beta oxidation. Fatty acyl-CoA molecules can move into the intermembrane space, but they cannot cross into the mitochondrial matrix. So once they are in the intermembrane space, they are converted into structures called acylcarnitines. The enzyme that carries out this conversion is called carnitine palmitoyl transferase 1, or CPT1. This is a membrane-bound enzyme that uses carnitine to essentially bump off the CoA portion of the fatty acyl-CoA molecule and create an acylcarnitine, which is a long chain of carbons and hydrogens, that's the R group, bonded to a C double bond O to a carnitine molecule. Now note that CPT1 is a membrane-bound enzyme, so in the cytosol, malonyl-CoA can inhibit this enzyme, and that's important because, as we talked about before, you don't want beta oxidation and fatty acid synthesis occurring at the same time. So when fatty acid synthesis is occurring, malonyl-CoA levels will get high. That will inhibit CPT1. There will be less carnitine shuttle activity and therefore less beta oxidation. Acylcarnitine molecules can then move into the mitochondrial matrix, and once you have acylcarnitine molecules in the mitochondrial matrix, they are converted back into fatty acyl-CoA molecules by an enzyme called CPT2. And once you have fatty acyl-CoA in the mitochondrial matrix, these molecules can now undergo the further steps in beta oxidation. So the big picture here is that the carnitine shuttle takes molecules of fatty acyl-CoA and moves them through this entire process into the mitochondrial matrix. If you are deficient in carnitine, you will be unable to metabolize fatty acids, and there are several potential reasons for which patients can become carnitine deficient. There are many secondary causes. If you are malnourished or if you have liver disease, you can develop a carnitine deficiency. In addition, patients who have high requirements for carnitine because they are performing lots of metabolism can become relatively deficient in carnitine. So these are patients who have had major trauma or burns, or sometimes women who are pregnant. And then finally, hemodialysis for patients with kidney disease removes carnitine. There's less synthesis of carnitine for unclear reasons in dialysis patients, and there's also a loss of carnitine through the membranes. So dialysis patients can become deficient in carnitine. The major consequence is the inability to transport long-chain fatty acids to the mitochondria. As we just saw, carnitine is part of the carnitine shuttle that moves these long-chain fatty acid molecules into the mitochondria. As a result, you get accumulation of long-chain fatty acids inside of cells. In addition, the diagnostic hallmarks of carnitine deficiencies are low serum levels of carnitine and acylcarnitine. If we go back a slide, if you're deficient in carnitine, you obviously will have a low level of carnitine because there isn't enough inside of your body. But in addition, because this level is low, you will have a low level of acylcarnitines because you are not able to combine carnitine with fatty acyl-CoA molecules. The symptoms of a carnitine deficiency include muscle weakness, especially during exercise. During exercise, you need to mobilize fatty acids as a source of energy for your muscles, and you won't be able to do that if you're deficient in carnitine. You can also potentially develop a cardiomyopathy because the heart also depends on fatty acids as a source of energy. And then another classic hallmark of a carnitine deficiency is hypoketotic hypoglycemia when fasting. So when you are fasting, you normally use fatty acids and glucose from gluconeogenesis as a source of energy. However, people with a carnitine deficiency cannot metabolize fatty acids, so they can only use glucose. They will overuse the glucose, and this will make them hypoglycemic. In addition, without fatty acid breakdown to generate acetyl-CoA, you do not have a source from which to generate ketones, so you have poor ketone synthesis without fatty acid breakdown. As a result, you get hypoketotic hypoglycemia, and this is a very unusual finding. Normally, when you are hypoglycemic, you also have high levels of ketone bodies because you're metabolizing lots of fatty acids. That is not the case in a carnitine deficiency. There's a rare inborn error of metabolism called a primary systemic carnitine deficiency. Babies born with this disorder have a mutation affecting carnitine uptake into cells. It's not that they lack carnitine or are deficient in carnitine, it's that they can't move the carnitine into their cells so that they can use it in the metabolism of fatty acids. Babies born with the infantile phenotype present in the first 2 years of life with a constellation of symptoms that are all related to buildup of fatty acids in the body that can't be metabolized. These babies often have encephalopathy and hepatomegaly. In addition, they can have high levels of ammonia from liver failure. They get hypoketotic hypoglycemia, as we discussed before. They also get low serum carnitine levels, and this may seem counterintuitive since I told you the problem is not carnitine deficiency. However, because the kidneys cannot resorb carnitine, it spills out in the urine and they become carnitine deficient. In addition, you find reduced carnitine levels in biopsy samples of the muscle, the liver, and the heart, all because the carnitine cannot be moved into cells. So back to our steps of beta oxidation. Now that we have moved our acyl-CoA molecules into the mitochondrial matrix, we can do the next step, which is to begin cycles of beta oxidation. Each cycle of beta oxidation will remove 2 carbons and shorten the chain by 2, and in the process it will generate NADH and FADH2 and acetyl-CoA. If you look at this acyl-CoA molecule I've shown on the bottom of the screen here, the alpha carbon is the carbon right next to the C bonded to O group, and the beta carbon is the carbon next to that. Basically, this process will lop off this molecule, and you may not realize this, but this is an acetyl-CoA molecule right here once you lop it off. It will then oxidize the beta carbon to attach an oxygen here. That's how it gets its name, beta oxidation. And then it can reattach the CoA group, and thus it will have shortened the fatty acid molecule by 2 carbons. The first step in a cycle of beta oxidation involves an enzyme called acyl-CoA dehydrogenase. This enzyme adds a double bond between the alpha and beta carbons. I've shown that here with a single bond between the 2 carbons, and after metabolism by acyl-CoA dehydrogenase, you now have a double bond, and in the process you generate a molecule of FADH2. There are many different acyl-CoA dehydrogenase enzymes that metabolize fatty acids based on the length of their chain. It's a family of 4 enzymes, and there's one that works on the short chain fatty acids, another one for the medium and long chain, and then finally a dehydrogenase that works on the very long chain fatty acids. And there's a well-described deficiency of the medium chain enzyme that you should know about for step 1. An MCAD deficiency is a medium chain acyl-CoA dehydrogenase deficiency. This is an autosomal recessive disorder that results in poor oxidation of 6 to 10 carbon fatty acids. If we go back a couple of slides, this is the enzyme deficiency in an MCAD disorder. As a result, you cannot lop off acetyl-CoA molecules from the end of these medium chain fatty acyl-CoA structures. If we go back to this slide of the carnitine shuttle, when you have an MCAD deficiency, you cannot perform further metabolism of fatty acyl-CoA. As a result, you will get high levels of acyl-carnitine in the plasma, and this is one of the hallmarks of an MCAD deficiency. So children with this deficiency have severe hypoglycemia without ketones during periods of fasting. They overuse glucose because they cannot metabolize fatty acids, and because they cannot metabolize fatty acids, they do not generate ketones well. Another unique finding of an MCAD deficiency is dicarboxylic acids of 6 to 10 carbons in length in the urine. This is the structure of a dicarboxylic acid here. These are synthesized by omega oxidation of fatty acids. Normally the beta carbon is oxidized. However, there is a minor pathway called omega fatty acid oxidation that oxidizes the omega carbon, which is the last carbon in the chain. Normally this pathway doesn't do much, but when beta oxidation is not functional, fatty acids will undergo omega oxidation. The result is production of dicarboxylic acids, and elevated levels of dicarboxylic acids are a hallmark of MCAD deficiency or any other process that disrupts beta oxidation. And then, as I said before, the levels of acyl-carnitine can become elevated because the carnitine shuttle is backed up. As I mentioned, there is severe hypoglycemia when fasting, and the reason for this is because gluconeogenesis is shut down. Pyruvate carboxylase, which is a key enzyme for gluconeogenesis, depends on acetyl-CoA, but acetyl-CoA levels get very low if you cannot perform beta oxidation. As a result, you have poor synthesis of glucose by the liver via gluconeogenesis, and in addition, all your other tissues are gobbling up the glucose because that is the only fuel available. This is all exacerbated by fasting, as I said before, and also in settings of infection when metabolic processes increase. And the treatment for children with this deficiency is to avoid fasting because it can be life-threatening. The last topic I'll discuss in this module is odd-chain fatty acid metabolism. When fatty acids have an even number of carbons, like 12 or 16 or 20, they are simply metabolized by beta oxidation until the molecule is gone. However, if the fatty acid has an odd number of carbons, beta oxidation will proceed until 3 carbons remain. When 3 carbons remain, you have a structure called propionyl-CoA that I've shown on the screen here. This is 1 carbon, this is a 2nd carbon, and this is the 3rd carbon. Propionyl-CoA will be metabolized into succinyl-CoA, which can enter the TCA cycle. And this is an important point because what this means is that odd-chain fatty acids can be used in gluconeogenesis to generate glucose. That is not true of even-chain fatty acids. They are metabolized to acetyl-CoA, which can enter the TCA cycle for energy, but cannot be used to generate glucose. And I've summarized that point here. Odd-chain fatty acids can lead to glucose. The first enzyme in the process of converting propionyl-CoA to succinyl-CoA is called propionyl-CoA carboxylase, and this requires biotin as a cofactor, just like all carboxylase enzymes. You should be aware that propionyl-CoA is a gateway to the TCA cycle for many different substances. We just talked about how odd-chain fatty acids can be converted to propionyl-CoA, but so can cholesterol and so can some amino acids like isoleucine, valine, threonine, and methionine. All of these structures here can be converted into propionyl-CoA and then into methylmalonyl-CoA using that carboxylase enzyme that requires biotin. From methylmalonyl-CoA, they can be converted to succinyl-CoA, and this reaction depends on vitamin B12. And then succinyl-CoA can enter the TCA cycle. This is also a nice place to point out that elevated levels of methylmalonic acid are seen in B12 deficiency, and that should make sense to you after looking at this slide. When B12 is deficient, you cannot convert methylmalonyl-CoA into succinyl-CoA, and then you can measure in the plasma the high levels of the methylmalonyl component of this molecule, which is called methylmalonic acid, and that is a hallmark of the B12 deficiency. There's a rare metabolic disorder called methylmalonic acidemia. This occurs when babies are born deficient in the enzyme methylmalonyl-CoA mutase. Methylmalonyl-CoA mutase catalyzes the conversion of methylmalonyl-CoA into succinyl-CoA. This is the reaction we were talking about on the last slide that is dependent on vitamin B12. Methylmalonyl-CoA and succinyl-CoA are actually the same structure in terms of their molecular makeup. The molecules are just arranged differently. They are isomers of one another, and so this enzyme catalyzes the isomerization reaction that converts methylmalonyl-CoA into succinyl-CoA. Babies who are born deficient in this enzyme will see their levels of methylmalonyl-CoA rise, and then the methylmalonate portion can dissociate, and you can have high levels of methylmalonic acid in the blood. Those high levels of methylmalonic acid can cause an anion gap metabolic acidosis. This can lead to CNS dysfunction, and this is a very serious condition. It's often fatal early in life. And that concludes our module on fatty acids.